Hello friends, welcome back to my YouTube channel Nesting Padasale. So in this video, we are going to do a last minute revision series for DBT BITP examination. So first is plant secondary metabolite, which are classified into 4 types based on the unit, the functional unit which they have. Terpene, the functional unit of terpene is they will be having an isoprene structure, which is a 5 carbon containing compound. And phenolics, they will be having a phenol ring, which means they will be having a benzene ring, and this phenolic they will be having a hydroxyl group with them. So if there is a question asking the plant secondary metabolite which have OH group with them, you should know it is phenolics. Okay. And next, nitrogen containing compound and sulfur containing compound. So terpenes are classified into monoterpene, sesquiterpene, diterpene, tetraterpene based on the isoprene unit that they have. Phenolics are again classified into coumarin, furanocoumaric, lignin, flavonoid, isoflavonoid, and tannin. So please read the example of each group of terpenes and each type of phenolics. Next is nitrogen containing compound include alkaloid. The example of alkaloid is cocaine, and cyanogenic glucoside. The example is purine, and non-protein amino acid, which is example is cannabinol. Okay, and sulfur containing compound include glutathione, glucosinolate, phytolaxin, threonine, dephesinalanine. Okay, so these are the plant derived terpenes which include hemiterpene which have just one isoprene unit, monoterpene they will be having 2, and sesquiterpene they will be having 3, and diterpene they will be having 4, and triterpene they will be having 5, and tetraterpene they will be having 8, and polyterpene they will be having more than 9 isoprene unit. Okay, and the example of monoterpene include your abscisic acid which is a plant growth hormone. An example of diterpene include vitamin A and gibberellic acid and azadirachtin. Okay, so this vitamin A is employed as an antioxidant, and gibberellic acid is again a plant growth hormone. So you should know abscisic acid is coming in a type called monoterpene, which means they will be having 2 isoprene unit and they will be having 10 carbon compound. Okay, whereas if you take diterpene, the example include vitamin E and gibberellic acid is coming in a diterpene. Okay, this particular azadirachtin, which is isolated, which you can find from the neem plant, it is used as an antifeedant. At the same time, it is also employed as an insecticide in various agriculture issues. Okay, so they will be disrupting the oviposition of various pesticide insects. Okay, next is pest causing insects. Okay, next is triterpene. The example is squalene. So this triterpene, again I'm telling you, they'll be having 5 isoprene unit, and this squalene is employed as a UV protector. And tetraterpene, the example is carotene, and polyterpene, the example is rubber. Okay. Next, plant derived alkaloids include atropine, which is used as an anticholinergic drug, and it is isolated from the Solanaceae family. Next is nicotine, which is actually employed as a potent poison, which is also again isolated from the Solanaceae family. Okay, and morphine, which is employed as a narcotic and anesthetic, which is isolated from Papaver family. Okay, so these atropine, nicotine, morphine are considered as a true alkaloid, and protoalkaloid include mescaline, hordenine, epinephrine. Okay, and pseudoalkaloid it include aconitine, threobromine, and coniine. Okay, so these are coming under pseudoalkaloid. Okay, and mescaline is employed as a hallucinogen which is isolated from the plants belonging to Cactaceae family, and other thing you can just give a read of yourselves. Okay. So the next thing we are going to study, and it is a highly important thing that you should study, permeability of phospholipid bilayer. So we all know lipid bilayer is selectively permeable. Okay. So you'll be seeing what are the compounds that can easily pass through the lipid bilayer and what are the compounds that are highly impermeable. Okay. So first, gases, they can easily get into the cell. Okay. They are permeable. Next is small uncharged polar molecule like ethanol, urea, water. They are also permeable, in which water is slightly permeable. So water need a water channel called aquaporin to get into the cell. Okay. Next is large uncharged polar molecule like glucose, fructose. They are impermeable. They can't able to enter into the cell without any kind of transport proteins, that they require the help of channel protein or carrier protein for their transport inside the cell. Ions also, they are impermeable. Next, charged polar molecule like amino acid, ATP, glucose 6-phosphate, protein, nucleic acid. They are also impermeable. Next, the mechanism of transporting ion and small molecule across cell membrane. So there are 4 different transport mechanisms are there: passive diffusion, facilitated diffusion, active transport, co-transport. Okay, so among this, which require protein? So only passive diffusion, they don't require any kind of protein for doing their activity, whereas facilitated diffusion, active transport, co-transport, they require few specific protein for doing their activity. Next, solute transported against this gradient. Okay, so only in active transport and in co-transport, solute molecules are transported against their concentration gradient. Okay. Next, only this active transport alone require the help of the ATP hydrolysis, whereas passive diffusion, facilitated diffusion, co-transport, they don't need any ATP hydrolysis for doing their activity. Okay. Next, only this co-transport alone is driven by the movement of co-transported ion down its concentration gradient. Okay. So oxygen, carbon dioxide, steroid, hormone, and many drugs, they move into the cell by this passive diffusion, whereas glucose, amino acid, ions, and waters are transported inside the cell by facilitated diffusion. Next is ions, small hydrophilic molecule, and lipid, they can enter into the cell by this active transport. Next, glucose and amino acid and various ions and sucrose, okay, they will be entering into the cell by co-transport mechanism. Okay, next in absorption in small intestine. So your vitamins, dietary lipids are entered, they can be absorbed by a process called simple diffusion. As your protein, vitamin B12 are absorbed by active transport, and fructose, they can move into the small intestine by facilitated diffusion, and monosaccharides are absorbed both by the facilitated diffusion and active transport, and glucose and galactose, they are transported into the cell by the secondary active transport mechanism. Next, those organelles with single membrane. Okay, so single membrane organelles include lysosomes, vacuoles, Golgi bodies, endoplasmic reticulum, and your microbodies like peroxisome, glyoxysome, glycosome, hydrogenosomes. Okay, they are covered with a single layer. Okay, next, double membrane bound organelles include mitochondria, chloroplast, nucleus, and plastid. So you should note endoplasmic reticulum is made up of single membrane, whereas mitochondria is made up of double membrane, inner membrane and outer membrane. Okay, so few organelles, they are called as non-membrane bound organelles. They don't have any membrane around them. This include ribosome, centromere, inclusion body, nucleolus, cilia, flagella, microtubule, microvilli, cytoskeleton, and centrioles. Okay. Next is autoimmune disease. Autoimmune diseases are classified into organ-specific and system-specific autoimmune diseases. Okay, so the example of organ-specific autoimmune disease include Addison disease, autoimmune hemolytic anemia, Goodpasture syndrome, Graves disease, Hashimoto thyroiditis, idiopathic thrombocytopenia purpura, insulin-dependent diabetes mellitus, myasthenia gravis, myocardial infarction, Peyronie's anemia, Streptococcus glomerulonephritis, spontaneous inflammation. They are coming under organ-specific category, and systemic autoimmune disease include ankylosing spondylitis, multiple sclerosis, rheumatoid arthritis, scleroderma, Sjögren's syndrome, systemic lupus erythematosus. Okay, so if you take Addison disease, the self-antigen will be present in the adrenal cells. Okay, that is specifically in adrenal medulla. Okay, next, autoimmune hemolytic anemia, self-antigen will be present on the RBC membrane protein, and in Goodpasture syndrome, the self-antigen will be present on renal basement membrane and lung basement membrane. In Graves disease, self-antigen will be present in thyroid stimulating hormone receptor. In Hashimoto thyroiditis, the self-antigen will be present in the thyroid protein and thyrocytes. Okay. Next, in insulin-dependent diabetes mellitus, the self-antigen will be present in the pancreatic beta cell, and in myasthenia gravis, the self-antigen will be present in the acetylcholine receptors. Okay. And in Peyronie's anemia, the self-antigen here it will be present in the gastric beta cells or interesting factor. Okay. And in spontaneous infertility, the self-antigen will be present in the sperm cells. Okay. Next, coming to ankylosing spondylitis, in which self-antigen will be present in your vertebrae, and multiple sclerosis. So myasthenia gravis is coming under organ specific, and multiple sclerosis is systemic autoimmune disorder. Okay, so multiple sclerosis, a self-antigen will be present in your brain, that is a white matter of brain. Okay, and rheumatoid arthritis, self-antigen is your IgG antibody, and sometimes self-antigen will be present in the connective tissue. Okay. Next, in Sjögren's syndrome, the self-antigen will be present in the liver, kidney, thyroid, sometimes in salivary gland. Next, in systemic lupus erythematosus, self-antigen might be your DNA, nuclear protein, RBC membrane, or platelet membrane. Okay, self-antigen will be present either in RBC membrane or platelet membrane. Okay, so this is also a very important topic. Next thing is regarding to antibiotic. Okay, so the cycloserine, vancomycin, bacitracin, penicillin, cephalosporin are called as cell wall synthesis inhibitor. Next is thymethoprim and sulfamides. They are called as anti-folate metabolite drugs. Okay. So they will be acting on, they will be disrupting the folate acid metabolism. Next is polymyxin, daptomycin. They will be inhibiting the cytoplasmic membrane structure. Okay. Next is with regarding to quinolines like nalidic acid, ciprofloxacin, ovobiacin. They will be inhibiting the process of DNA replication by attacking the DNA gyrase enzyme and topoisomerase. Next is actinomycin. This actinomycin, rifampicin, streptovirus are called as transcription inhibitor. Okay, actinomycin they inhibit the process of RNA elongation, and this rifamycin, streptovirus they stop the transcription process by binding to DNA dependent RNA polymerases. Okay, next is erythromycin, chloramphenicol, clindamycin, lingomycin. They inhibit the translation process, but all the four antibiotic they will be inhibit the process by attaching to 50A subunit of ribosome. Whereas tetracycline, spectinomycin, streptomycin, gentamicin, kanamycin, amikacin, nitrofurans, they too inhibit the translation, but they will be attacking the 30th subunit. Okay. Next is mupirocin and puromycin. They will be inhibiting the translation by acting on the transfer RNA synthesis, and they will be disrupting the mechanism. Okay. Next, derivative from primary germ layer. So primary germ layer is classified into ectoderm, mesoderm, endoderm. If you take ectoderm, all the nervous tissue and the epidermis of skin and epidermal derivatives, a cornea, lens of eye, epithelium of oral cavity, nasal cavity, anal canal, and tooth enamel, epithelium of pineal gland, pituitary gland, and adrenal medulla, and melanocyte, and some cranial bodies will be derived from the ectoderm. So from mesoderm, your skeletal muscle, smooth muscle, cardiac muscle, blood, bone marrow, lymphoid tissue, endothelium of blood vessel lymphatics, synovial membrane of joint cavities, and organelles of urinogenital syndrome like ureter, kidney, gonads, reproductive ducts. So you should know reproductive ducts, kidney, gonads are derived from the mesoderm. Next is endoderm. So from endoderm, your epithelium of digestive tract except that of oral cavity and anal cavity. So your epithelium of oral cavity and anal cavity are derived from ectoderm, whereas from this endoderm, the epithelium of digestive tract is being produced, and glandular derivatives of digestive tract is produced from the endoderm. Okay, next, epithelium of your respiratory tract and other auditory tube and tonsils are derived from endoderm. Thyroid, parathyroid, thymus gland, epithelium of reproductive ducts gland, epithelium of urethra, bladder, or urethra and bladder are derived from endoderm. Okay. So the next important thing is cleavage pattern and eggs. Okay. So if you take echinoderms and amphioxus, they have holoblastic radial cleavage and egg is isolexical. Okay. Next, annelids, mollusk and flatworm. They have holoblastic spiral cleavage, and tunicate they have holoblastic bilateral cleavage. Next, mammals, nematode, they have holoblastic rotational cleavage. Amphibians, they have holoblastic radial cleavage. Cephalopods, they have meroblastic bilateral cleavage. Okay, so tunicate they have holoblastic bilateral cleavage, and cephalopods they have meroblastic bilateral cleavage, and fish, reptile, birds they have meroblastic discoidal cleavage. Insects they have meroblastic and superficial cleavage. Okay, and insects the central is the legs is present. Okay, so insects have meroblastic superficial cleavage pattern. Next, gastrulation movement. So first is invagination, which is an infolding of infolding of epithelium into the embryo, and the type of movement which you can find in sea urchin endoderm. Okay, next is involution. It is an inward movement of expanding outer layer of cell so that it will be spreading over the inner surface of remaining external cell. Okay, so the cell that are expanding will start to go inner side so that they will be forming a second layer. Okay, example is amphibian mesoderm. Next is ingression, where there will be a migration of individual cell from the surface into the embryo interior. Example includes sea urchin mesoderm, drosophila neuroblast cell. They will be forming by undergoing gastrulation movement named ingression. Next is delamination, which is a splitting of one sheet into a two nearly parallel sheet. Example include hyphoblast formation in mammals and birds. Next is epiboly, which is an expansion of one sheet cell over the other. So don't confuse with involution and epiboly. The example of epiboly include ectoderm formation, which is an outer layer in amphibian, sea urchin and tunicates. Okay. Next is some common inherited disorders. So first is autosomal recessive pattern disorder includes sickle cell anemia, cystic fibrosis, phenylketonuria, Tay Sachs disease. They are having a autosomal recessive pattern. And sickle cell anemia, there will be a abnormal hemoglobin which will be causing a disformation of your RBC cell. At the same time, these sickle cell anemia patient, they will be having a resistance towards malaria. Next is cystic fibrosis, in which there will be a defective chloride channel which is responsible for transporting the chloride ion, which will will be ultimately lead to excessive mucus formation in lungs. Next is phenylketonuria, which is an inborn error of amino acid metabolism, where in this particular disease case there will be a defective enzyme in phenylalanine metabolism. The defective enzyme is tyrosine hydroxylase. Sorry, I'm just pronouncing them wrong. Tyrosine hydroxylase. Okay, which will be resulting in excessive amount of phenylalanine will be started to deposit, which will be ultimately lead to mental retardation. Okay, next is Tay Sachs disease, in which there will be a defecting hexamidinase enzyme. Okay, so this enzyme will be defective, so there will be excessive accumulation of sphingolipids in the lysosomes of neurons, thereby implicating the neural development. Next is autosomal dominant disease pattern include Huntington disease, where there will be a defective neural protein called Huntington, which will be started to assemble into aggregates, which ultimately lead to damage in the neural tissue. Next, in hypercholesterolemia, there will be a defective LDL receptor, which will be leading to the production of excessive amount of cholesterol in your blood, which is ultimately leading to early heart attack. Next, X-linked recessive pattern include DMD, in which there will be a defective dystrophin protein. Dystrophin is a cytoskeletal protein, so ultimately it will be leading to impairment in the muscle function. The last is hemophilia, which is caused by there will be defective blood clotting factor 8, so it will be ultimately leading to an uncontrolled bleeding. Okay, so the next is DNA damage repair system. Okay, so mismatch repair, they will be correcting replication errors, and the enzyme involved include mutase, mutal, and mutH in case of E. coli. Next, photoreactivation, they will be correcting pyrimidine dimers, and the enzyme involved in photoreactivation is DNA photolyase. And next is base excision repair. It will be removing the damaged base, and the enzyme involved is DNA glycosylase. Okay. Next is nucleotide excision repair, which will be removing your pyrimidine dimer, and it also remove the bulky adduct on base. Okay. And the enzyme or other proteins involved include UVR A, B, C, D in case of E. coli. And double standard break repair, they will be correcting double standard break, and rect A and rect B, C, D in E. coli, or the enzymes that are involved in double standard break repair system. And translations DNA synthesis, which will be correcting pyrimidine dimer, it will be correcting A purinic site, or it will be removing the bulky adduct on base. Okay, so the Y family DNA polymerase like UMUC in E. coli, they will be catalyzing the process of translation DNA synthesis. Okay, so sometimes there will be some human diseases will be arising if this particular DNA repair system is not removing the damage that is caused by some other factor. So DNA mismatch repair system, if it is not been active or if it is disrupted, means then it will be lead to the formation of hereditary non-polyps colorectal cancer. Okay, so this DNA repair system will be correcting the damage that is caused by UV radiation and chemical mutagenesis. Okay, so the person will be having a cancer susceptibility in colon and ovary. Okay, next is xeroderma pigmentosum, which is arise due to the defect in nucleotide excision repair system. And this particular nucleotide excision repair system, they will be removing the damage caused by UV radiation and point mutation. So the cancer susceptibility itself, they will be having a increased susceptibility to get skin carcinoma and melanoma. Okay, next this Bloom syndrome, Fanconi anemia, and hereditary breast cancer, they are caused due to the repair of double standard break. Okay, so if this particular repair system is not been activating, not been functioning, then it will be occurring this particular thing. Okay, so next thing is regarding to ABO blood group. If you take A blood group, they will be having antigen A in their RBC and anti-A antibody in their serum. Okay. Next, B blood group, they will be having B antigen on their RBC and they will be having anti-B on their serum. Okay. In their serum. Okay. Next, ABO blood group system, they will be having both anti-A and anti-B on the surface of RBC, but they don't have any serum antigen in their blood. Next is O blood group, they don't have any antigen in their RBC, okay, but they have both anti-A and anti-B antibody in their serum, okay. Next, the selector molecular mission. So we know in our body, in each cell, there will be different molecular missions are in there. So this is an important topic. So if you take ribosome, it has helicase, primase, DNA polymerase, and it is located in nucleus. So they are generally involved in process of DNA replication. Next is transcription initiation complex. They have promoter binding protein, helicase, transcription factor. They have RNA polymerase. Okay, they are also present in nucleus and they are involved in RNA synthesis. Next is spliceosome. So spliceosome, they have pre-mRNA, small nuclear RNA, and they also have protein factor, and they are also located in nucleus and they are involving in mRNA splicing process. Next is nucleopore complex, which they have nucleoporins with them. They are present in nuclear membrane, and the main function of this nuclear pore complex is that they are involved in nuclear export and import. Okay. Next is ribosome, where they have ribosomal protein and they have 4 ribosomal RNA molecule which are organized in larger subunit and smaller subunit. Additionally, they have other protein factor. Okay. So these ribosomes are present in cytoplasm and endoplasmic reticulum membrane, and they are involved in process of protein synthesis. Next is chaperone. So the main component in case of bacteria include GRO-EL and GRO-ES. They are present in cytoplasm, mitochondria and ER. They are involving the process of protein folding. Next is proteasome, which have core protein, regulated protein in them, and they are generally present in cytoplasm, and the main function of this proteasome is that they are involving in protein degradation. Next is photosystem. Okay, so they are involved in the process of photosynthesis, and this photosystem, they are light harvesting. They have light harvesting complex and they have reaction center with them. So this light harvesting compound, they will be having a multiple proteins and pigment to harvest the light, and the reaction center, they have multi subunit protein which are generally associated with the pigments and electron carrier. Okay, this photosystem, they are located in the thylakoid membrane in plant chloroplast, and in case of photosynthetic bacteria, they will be present in the plasma membrane. Okay, next is MAP kinase cascades, which have scaffold protein and some other protein kinases with them, and they are present in cytoplasm, and this MAP kinase are involved in the process of signal transduction. Next is sarcomere, which include thick filament, that is thick myosin filament, thin actin filament, Z-line, tintin, nebulin. They are involved, they are present in the cytoplasm of muscle cells or myocytes. They are involved in the process of contraction. Next, we are going to study the leading therapeutic proteins in Indian market. So first is factor 8, which is expressed in Chinese hamster ovary cells, and they are employed for hemophilia disease. Next is streptokinase, which is employed for acute myocardial infarction, and it is from E. coli. Next is tissue plasminogen activator. Again, it is also employed for acute myocardial infarction, and they are expressed in both in E. coli as well as Chinese hamster ovary cell. Next is hormone that include insulin, which is employed for diabetes condition, and it is expressed in E. coli. Next is erythropoietin, which is employed for anemia, and it is employed in mammalian cell line. Okay, next is human growth hormone, which is employed for human growth hormone deficiency in children. Okay, and they are expressed in E. coli. Okay, next is alpha interferon, which is expressed in E. coli, and it is employed for cancer, hepatitis and leukemia. Next is beta interferon, which is again expressed in both in Chinese hamster ovary cell and E. coli. They are employed for curing sclerosis, multiple sclerosis. Next is gamma interferon, which is expressed only in E. coli, and it is used for chronic glaucomatosis disease. Okay. Next is interleukin 2, which is expressed in E. coli, and it is employed for renal cell carcinoma. Next is hepatitis B, which is expressed in Saccharomyces cerevisiae. It is generally employed for preventing the hepatitis B infection. Okay, next we will be seeing the antiviral drug, the viral disease that they are used on the target. So if you take herpes virus, the vidarabine is employed, and they will be targeting the virus polymerase enzyme. Next is herpes simplex virus. The drug employed is acyclovir, which is also targeting virus polymer. Next is cytomegalovirus, where they will be using a drug named ganciclovir. They will be also targeting a viral polymerase. And next is retrovirus. So retroviral drugs are classified into two types based on the target. If you take zidovudine and lamivudine, they will be targeting the reverse transcriptase enzyme, and if you take this sacunavar, nelfinavar, indinavar and ritonavir, navir, they will be targeting the HIV protease enzyme. Okay. Next is influenza A virus. They will be employing the amantadine and rimatadine. Okay. So they will be targeting hemagglutinin protein, and for influenza B, they are employing this 2-duct Tamiflu, which will be acting as a neuraminidase inhibitor. Okay. Next, for pipcona virus, they are incorporating this particular drug called peliconaril, which will be blocking the attachment of viral into the host cell. They will be blocking the attachment, so viral can't able to attach to the host cell. Okay. Next, we'll be seeing the human therapeutic protein obtained from plant. So first protein is protein C, which is obtained from the plant called tobacco, and it is employed as an anticoagulant. Next is hirudine, which is isolated from canola, and it is employed as an thrombin inhibitor. Okay. Next is granulocyte macrophage colony stimulated factor, which is also again produced from tobacco, and it is employed for the condition called neutropenia. Next, somatotrophin, it is isolated from tobacco, it is employed for growth hormone, it is employed as a growth hormone. Next is erythropoietin, which is also isolated from the tobacco, and it is employed for anemia disease. And next is this particular enkephalin, which is isolated from arabidosis, and it is employed as a painkiller because it is employed as an anti-hyperanalgesic drug due to the opiate activity of arabidosis. Okay. Next is epidermal growth factor, which is also isolated from tobacco. It is also employed for control of cell proliferation, and it is additionally used for wound repair mechanism. Next is interferon alpha, which is isolated. Interferon generally isolated from rice, turnip, and tobacco. They are employed for the disease like hepatitis C, hepatitis B. Okay. Next is serum albumin, which is isolated from tobacco. It is used for burn, surgery, and liver cirrhosis. Okay. Next is hemoglobin alpha and beta chain, where they are also isolated from tobacco. So most of the therapeutic proteins are isolated from tobacco. And coming back to hemoglobin, they are isolated from tobacco, which is also used as a blood constituent. Next is this particular homotrimeric collagen, which is also isolated from tobacco, and it is also employed as an alternative for collagen. Next is alpha 1 antitrypsin trypsin inhibitor, which is isolated from rice. So the alpha 1 antitrypsin trypsin inhibitor is from rice. It is used for cystic fibrosis disease curing, and also it is used for treatment of liver disease. Next is aprogenine, which is isolated from maize plant, and it is used in transplant surgery. And next is lactoferrin, which is from potato. It is employed as an antimicrobial. Next, we'll be seeing the properties of restriction endonuclease. So, restriction endonuclease are classified into three types. So, first, with regarding to type 1 restriction enzyme, they have a single multifunctional enzyme carrying out the process of restriction and modification. Whereas, if you take type 2, they have a separate subunit for nuclease activity and methylase activity. Okay. Next, type 3 enzyme, they have a separate enzyme unit will be there. Both the enzymes, they will be sharing a common subunit. Okay. Next, with regarding to nuclease subunit structure, type 1 they are called heterotrimer, type 2 they are homodimer, and type 3 they are heterodimer. Okay. The cofactor requirement, type 1 they require ATP, magnesium ion, and S-adenosine methionine, whereas type 2 they require only magnesium ion, and type 3 also they require magnesium ion. Additionally, some will be requiring S-adenosine methionine. So type 1 restriction enzyme alone require ATP for them. Okay. Next is regarding to DNA cleavage requirement. Okay. So this type 1 they need 2 recognition site in any orientation, whereas type 2 restriction enzyme they have only single recognition site, and for type 3 restriction enzyme they need 2 recognition sites which should be located in head-to-head rotation. Okay. Next is head-to-head orientation will be there. Okay. So for type 1 there is no restriction. They can be 2 restriction sites. They might be in any orientation. But for type 3 they need 2 recognition sites. They should be oriented in head-to-head fashion. Okay. Next, site of DNA cleavage, type 1 will be cleaving randomly 1000 base pair away from the recognition site, and both the type 1 they will be cleaving at the recognition site or near the recognition site. Next, type 3 they will be cleaving approximately 24 to 26 base pair to the 3' side of the recognition site. Okay. So next is regarding to the recognition sequence. So first important thing you should know is that except this Proteus vulgarius, that is PVU2, and regarding to this Arthobacter fluidus, that is ALU1, and this Haemophilus aegyptius, that is HAE3, they will be producing blunt-end. Rest of after this 3, every restriction enzyme they will be producing sticky end only. Okay, so if you take eco R1, they are isolated from Escherichia coli, and the recognition sequence for this particular enzyme is GAATTC. Okay, so just know for this particular eco R1, and also for HIND3, it is producing sticky end, and the recognition site for HIND3 is AAGCTT. Okay, and SOR3 is isolated from, it's taken from Staphylococcus aureus, and ALU1 is from Arthobacter fluidus, and the STAG1 is from Thermus aquaticus. Okay, so next thing regarding to properties of DNA polymerase 1, 2, 3 from E. coli. So first is DNA polymerase 1, which is involving DNA repair and primer removal, and DNA poly 2 is involved only in DNA repair, and DNA poly 3 is involving DNA replication. Okay, and regarding to 5' through 3' polymerization activity, all the 3 enzymes they have this particular activity. Next is 3' to 5' exonuclease activity. It's also called as proofreading activity. So all the 3 enzymes they have this particular 3' to 5' endonuclease activity. They have proofreading activity. Next is 5' to 3' exonuclease activity, which is responsible for removing the primer in lagging strand synthesis. So this polymerase 1 alone having this 5' to 3' exonuclease activity. That's why this polymerase 1 is involving in removing the primer during the lagging strand synthesis. Next is general property of commonly used vector. The first is plasmid. They are used for general DNA manipulation, Regarding to lambda vector, insertion lambda vector are used for construction of cDNA libraries, and replacement lambda vector they are employed for construction of genomic libraries. Again, cosmids also employed for genomic libraries, whereas this M13 phage they are used for two applications. First application they are employed for in vitro mutagenesis, and next application they are used for DNA sequencing. Next is this phage which is used again for general DNA manipulation, and also it is employed for in vitro mutagenesis. Okay. Next is YAG, PAC, BAC. They are having a high cloning capacity, so they are employed for construction of genomic library. Okay. The most important thing you should know about the maximum insert size of each vector. If you take plasmid, the maximum insert size is 10 to 20 kb of DNA. They can be inserted or they can be cloned. Lambda insertion vector, the insertion size is approximately 10 kb, and lambda replacement vector, the approximate insertion size is approximately 23 kb, and for cosmid, the approximate insert size is 44, and for M13, the approximate insert size is 8 to 9 kb. And for phage mean the approximate insert size is 10 to 20. Whereas for YAG, the approximate insert sizes, YAG can able to accept the DNA ranging from 200 base pair, that is 200 kilo base pair to 2000 kilo base pair. Okay. And PAC, they can clone up to 75 kilo base pair to 90 kilo base pair. So this is I am telling with regarding to kilo base pair. Okay. BAC, they can clone the DNA segment ranging from 130 kilo base pair to 150 kilo base pair. Next, a principal type of RNA and their respective function. First is messenger RNA, which is coding for protein. Ribosomal RNA, they form a basic structure of ribosome. Transfer RNA, they are central to protein synthesis. This transfer RNA will be acting as an adapter between your mRNA and the amino acid. Next is small nuclear RNA, which will be functioning in various nuclear process. The small nuclear RNA involved in process of splicing of your pre-mRNA. Okay. Next is small nucleolar RNA, which are used to modify the ribosomal RNA, and the small Kajal RNA, which are employed for modifying both the SNO RNAs and snRNAs. Okay. Next is microRNA. This microRNA regulate the process of gene expression by blocking the translation of selective mRNA. Okay. Next is this siRNA, which is called a small interfering RNA. Again, the small interfering RNA will also turn on the gene expression by they will be directing the degradation of selective messenger RNA. Okay, and also they are involving in establishment of compact chromatin structures. Okay, the small interfering RNA are involving in establishment of compact chromatin structure. Okay, some other non-coding RNA are involving in the other process, like they are involved in telomere synthesis, they are involving in transport of protein into ER at the time of protein trafficking, and some are involved in X chromosome inactivation. Next, we'll be seeing about three major class of transposable element, which include DNA only transposon, retroviral transposon, retrotransposon, and non-retroviral retrotransposon. So first, if you take DNA only transposon, they have short inverted repeat at each end. The enzyme that is required is transposase. The example include P element of Drosophila, ACDC element of maize, TN3 and TN10 of E. coli, and TAM3 of Snapdragon. They are coming under Snapdragon. They are Snapdragon. They are coming under the class of DNA only transposon. Next is retroviral like retrotransposon. They will be having a directed repeat, long terminal repeats. They will be having this LTR at each end. They have reverse transcriptase and they have integrase function. They require both the reverse transcriptase function and integrase function. This example include copia element of Drosophila, TY1 element of yeast, THE element of human, and BS1 element of maize. Okay. Next is non-retroviral retrotransposon, which they will be having a poly A tail at their 3' end of the RNA transcript, and their 5' end is often truncated. They require reverse transcriptase and they also require endonuclease with them. The example of this non-retroviral retrotransposon include F1 element in Drosophila, N1 element in human, and CIN4 element in mice. Okay. Next is maize. Next is we are going to study few deviation from universal genetic code. So first code is UGA, which is coding for stop codon, but in Mycoplasma, Spiroplasma, mitochondria from many species, they are coding for tryptophan amino acid. Okay, next is CUG. In most of the animal, they will be coding for the amino acid leucine. But in mitochondria and yeast, they will be coding for the amino acid 309. Next is UAA and UAG, which will be coding for stop codon. They are considered stop codon, but in this particular acetabularia, tetrahymena and paramecium, they will be coding for the amino acid called glutamine. Next is UGA, which is again a stop codon, but in this particular protozoan euploids, they are coding for the cysteine amino acid. Cysteine amino acid, they will be coding. Okay. So in the next video, we'll be continuing with the rest of the important topics. So try to study in this particular 25 topics which I have listed over in this particular video. Thank you friends. Thanks for watching this video.